Assalamu alaikum and good evening all of you. Before starting anything, I would like to share a famous spiritual quote which I always use to amplify my abilities. It goes like Imam Ali Raza quote from his father Imam Musa Qasim. He quote from his father Imam Jafar Sadiq. He quote from his father Imam Muhammad Baqir. He quote from his father Imam Zainul Abidin. He quote from his father Imam Hussain. He quote from his father Maula Ali. He quote from the Holy Prophet Muhammad sallallahu alaihi wasallam. And he said, what faith is? Faith is knowing from your heart, expressing it through your tongue, and performing it with your organs. So whatever you do in life, faith is the basic necessity for everything. So proceeding towards our next example, in which we will discuss that how we can evaluate that modifications can be done in a drug in order to make it potent and increase its affinity towards the receptor. So this is our pocket in which 5 systems are present. This in black is our drug. So in solving this question, we will mark the drug components which can be modified, and we will mark them and locate them with the possible modifications. So starting from these two systems, this OH, that is hydroxyl, can interact with a proton donating group by consuming or by providing the lone pair of oxygen. So a proton donating ability should be present here. This is the first point. Secondly, to this negatively charged oxygen, a positively charged structure should be present. So we will mark this. So we have marked this R group and we have named it A, and to A we have written a quaternary nitrogen substitution should be done in order to increase the drug's potency and affinity. And I have drawn a quaternary nitrogen showing you that how this quaternary nitrogen can interact with the pocket. The quaternary nitrogen should be connected to 2 hydrogen, and with this bond it should be connected to this nitrogen. So 3 bonds are present. However, a 4th bond should also be present for making it quaternary. The hydrogens of these nitrogen, for example, this hydrogen will be in close proximity to the OH, that is, it will easily interact with the oxygen in the form of a hydrogen bond. And this nitrogen being positive and in close proximity to this oxygen can form ionic interaction with this negatively charged oxygen. So we marked the component of drug on which modification should be made, and we named it, and to that name we added that what substitution can be made. And for you I have drawn an example that how this modification can interact in real. Now coming towards this pocket which have a carbonyl, that is, this oxygen can interact with the drug component in a proton accepting way, but in our drug no proton donating component is present. So we will mark this structure. So we have marked this structure, this component of drug as B, and to B we have written that a proton donor is required, and the example which I have used is amine, and I have drawn an amine so that you can see that how the interaction can take place. Instead of this component, if an amine would be present, the oxygen can interact with this hydrogen in the form of hydrogen bonding through proton donor and acceptance phenomenon. So to B we have marked that a proton donor is required in order to make interaction, which means that a modification in the form of proton donor at this component would be efficient. Now coming towards the next system of the receptor, that is a carbonyl is present. This carbonyl contains an oxygen which have two lone pairs of electrons. The system near it should be this carbonyl, which can be modified in such a way that a proton donor should be present here, through which this oxygen can interact in the form of hydrogen bonding. So we will mark this component of drug and will write down that what modification is required. So we have marked this component of drug as C, and to C we have written a proton donor is required, and the example which I am using is again amine. So substituting an amine with this component of drug can increase the affinity of the drug to receptor in two ways. So I have drawn an amine, this is the nitrogen of amine, and these are the two hydrogens of amine. By substituting this amine with this component of the drug can interact with the receptor in two ways simultaneously. That is, one hydrogen of this amine would be involved in hydrogen bonding with this oxygen of the receptor, and this hydrogen of amine can interact with this oxygen of the receptor. So substituting an amine here in this component increases the affinity of the drug towards the receptor by two folds, or by two ways. That is, both the hydrogens of amine can interact with the oxygens of the receptor. So amine is the best modification in this component of the drug. So proceeding towards the next system of the drug, sorry for a mistake throughout this procedure. I didn't saw that to a nitrogen a cyclopentane ring was connected, which I saw now in the question. So I have added this cyclic ring structure in the component so that the complete question can be solved. So this component in this example cannot interact with the drug component, but however, if you can extend this chain in such a way that it should direct towards this cyclic structure, so in some way this lipophilic system can interact with the receptor's lipophilic system. So we will mark this component of drug and we will write it as D, and to D we will state that chain extension should be done in order to ensure that the drug interacts with this system of pocket as well. So I have marked this component of the drug as D, and to D I have stated that chain extension is required, and I have drawn a chain extension through which you can see that how chain extension can interact with the pocket system. So this is the chain extension. It can be extended in multiple ways, but the example I have used is a simple and short extension, that is an aromatic system which was present here is present here, and due to an aliphatic chain system, it is directed towards a cyclic pentane structure, or any system you can use. An aromatic ring can be used here, but I have used a cyclopentane structure which can interact with the cyclopentane structure of the component. So this was the final modifications. So we have evaluated 4 modifications through which the drug can interact with the receptor. The drug's affinity towards the receptor can be increased through these 4 modifications. So this is what we were required. The question was that how the drug can be modified in order to interact with the receptor and to increase its affinity. So through 4 modifications we have evaluated that how this drug or increase its affinity towards the receptor. So this was all about structure modifications according to the receptor pocket.